Hello there. Now, this is going to sound really familiar if you bothered to pay attention to the first chapter of grade 9 science. Why? Because in Canadian textbooks, and actually most North American textbooks really, in science class they will typically start with, oh, why do we do science? Why is it important? And honestly, in a lot of textbook series, it will actually be mostly copy-pasted. But there is in fact something new in grade 10 science for skills of scientific investigation. Now, what is it? Well, we'll get there in a moment. Now, as we well remember, I hope, if we don't, then great, this is a review. So as we well remember, there are two main types of scientific inquiry. Both of them have variables. What are variables? Well, they are capable of varying. They are able to vary, hence the name. Variables are things that can be different depending on what you do in the experiment or what you start with in the experiment. So wait a second. If we start somewhere in the experiment, because for one particular experiment we're supposed to follow the same procedure with different starting conditions to see what happens to the result. If we start with something, well, the independent variable is changed by the investigator. So, starting condition. And the dependent variable is the variable that changes in response to the independent variable. So it depends on the independent variable. Well, isn't that just result? Well, yes it is. So most of the time when we say dependent variable, we basically just mean result. Now you might think, hey, why is it that a variable is any condition that changes or varies the outcome of a scientific inquiry? Doesn't that sound only like independent variables? Well, no. Even though the dependent variable is your result, your actual outcome of your inquiry, i.e. the outcome of a question you ask, or a hypothesis that you're testing, an educated guess, so to speak, that you're testing, well, that depends on the dependent variable. Hence why this definition of variable is not in fact in conflict with the fact that, well, this is the result. Now, when you're actually conducting inquiries, you should make sure you start with different independent variables, follow the same procedure to make sure that you're testing what that particular procedure does, and then you get, well, your dependent variables. Now, there are two types of experiments. The first one is the one that you can actually do this sort of procedure in, which is a controlled experiment. What's the second type? Well, wait a second. Observational study. Why is it that we can't do controlled experiments on everything? Well, let's just say that manipulating, say, a tectonic activity is not exactly a very practical controlled experiment with our current technology level. Hence why there are many things that we can only study observationally and then draw conclusions from there. And if we can't even manipulate tectonic plates, then of course manipulating planetary orbits and such is far beyond our current technology level. Hopefully though one day we will get there. Now, a controlled experiment has the independent variable purposely changed. Now, if you've ever done a controlled experiment, which I hope you have, you may have heard of this thing called a control. The control is the exact same procedure, but the beginning is a certain condition. For example, when you are testing the effectiveness of a drug, the control group is usually the group that gets no treatment. Then there is a placebo group. Placebo. The placebo group gets pills, but the pills have no active ingredient. So this is a, hey, I think I took a drug so I feel better type. Actually, it is possible to think yourself into having the flu or having the cold. So it's not that unreasonable that they should test for a placebo group. And then there's the actual treatment group. However, even though controlled experiments, should always at least have control groups, that's not the reason why they're called a controlled experiment. They're called a controlled experiment because you control the independent variables, as opposed to observational studies where you just look at what's already there and you draw conclusions from there. So a controlled experiment can be run many times and is generally the cheaper of the two for what I hope are relatively obvious reasons, as in it's cheap enough and small enough that you can manipulate it repeatedly instead of just having to rely on, too expensive or too technically difficult to manipulate, and therefore we do an observational study. Now, there is a third type of investigation, so to speak. I mean, technically speaking, the correlational study is both, either one, really, or both, of the observational study and the controlled experiment. Because it takes the data that we have for two particular variables, and what does it do with these? Well, it looks to see if there's any trend between them, any correlation. So here we clearly see that as one variable gets bigger, the other variable also gets bigger. So this is a positive correlation. As one gets bigger, the other one gets smaller. That's a negative correlation. Now, if there's just randomness, then we have no correlation. So, we do note that correlations can vary in strength. For example, here, this is fairly tight. The maximum strength a correlation can have is 1, i.e. all the dots are exactly on the trend line. The minimum strength it can have is 0, which is there is no trend whatsoever. And here we see, there's some vague correlation, but it's really weak. So the strength of correlations is generally fairly indicative of how much the independent variable affects the dependent variable versus how much, say, outside factors affect it. For example, in biology there is a concept called phenotypic plasticity, often just abbreviated as plasticity. Now, when something deforms elastically, it bounces back to its original shape. When it deforms plastically, it stays in its new shape, or it can break, depending on properties. But plastic was essentially a term for a material that can be molded easily. So if the phenotype, which is the appearance of some organism, can be changed fairly easily, it is relatively plastic. So plasticity here means how much the phenotype can vary depending on environmental factors while having the same genotype, i.e. the same genes. So phenotypic plasticity is responsible for, in some cases of genetic diseases, both variable expressivity and variable penetrance. So we will not talk about those concepts for now because they are well beyond the scope of grade 10 science. Just know that the weak and strong correlations tend to relate to how much exactly does the independent variable dictate the dependent variable versus the effect of, say, other factors. So in biology, that's usually nature versus nurture, so to speak, in relatively layman's terms, i.e. average people. So, scientific investigation skills. Obviously if we have to do an investigation, we need to have some skills oriented toward it. Now, the first skill we need to do is to know what we are actually doing. Which means, Eisenhower once said, "I have found that plans are useless, but planning is essential." He said something along those lines, but the general idea is the same. So even though you might have to change your plans halfway through, or you often do if the results are not what you expect, you still have to plan. And first of all, you need to know what you're doing, which is initiating. So initiating and planning includes coming up with a hypothesis, i.e. an educated guess of what might be going on. So this is a possible answer. It can be tested. For example, if my cell phone doesn't work, I can test whether or not the boot up program is messed up, in which case I might have to do a hard reset, or if I should just plug in the charger and wait, i.e. the battery is dead. So if I test one option, i.e. the hard reset, and it doesn't work, well, okay, I know that's been falsified. That hypothesis is falsified. And if I plug in the battery and it still doesn't work, well, okay, that's falsified as well. Let's try something else. Get another phone, call support. All right. So besides that, those are several hypotheses that can be advanced as to why my cell phone doesn't work. Now, if I was to make a prediction based on a hypothesis, i.e., oh, it's a battery's fault, my prediction would be, after I plug it in and charge it for a while, I can turn it on. So the prediction is the outcome of the controlled experiment based on a hypothesis. Now, of course, after we make these plans, we have to actually do the experiment. And of course, observe what happens. I mean, okay, I did an experiment. What happened? I don't know. Obviously, that doesn't work, right? So there are quantitative, i.e. quantity. You have probably heard of quantity as in numbers. And yes, that's exactly what quantitative is. And qualitative, a quality of an object. So this object is red, this object is blue. These can be seen as qualitative, i.e. they're categories. Or this is square, that one is a triangle. Whereas quantitative, for say red, it would be the maximum absorption spectrum is at blah 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 blah. Or the spectrum of reflected light is this, or whatever diagram. That would be a quantitative counterpart to the qualitative observation that an object is red, or blue, or green. So a quantitative observation, numerical. Qualitative, non-numerical. Now, after you actually do the experiment and record your information, or record your results rather, of course you should record your results according to the independent variables that they began with, for what I hope are obvious reasons. Otherwise, how would you ever link your independent and dependent variables together? You wouldn't, which would be bad. So, afterward, you clearly need to actually look at your information, right? So, look at data, figure out what is going on. Figure out, well, actually, there's a few things that you can figure out. You can figure out what is actually going on, or you can just figure out whether or not it accepts or rejects your hypothesis. Any hypotheses. This is the plural of hypothesis. The singular form has an I instead of an E here. So after you've analyzed and interpreted the data, I figure out what is going on. What do you have to do next? I mean, obviously, if you don't write a report, you will not get the grades for that mark, for that particular lab. So if you don't get marks, well, that's not good, right? Same thing works in terms of journals. Publish or perish, they call it. Or perish. So this is the phrase I learned at university while doing lab work for their version of do or die. It is greatly longer, as you can tell. So in summary, We do science because we observe things in nature and we try to figure out what is going on. Or alternatively, we're curious about, hey, what will happen to this if I do this to it? So, for these reasons, we conduct, well, controlled experiments if it's a case of what happens if I do this to this. Or we can do observational studies in terms of, let me figure out what's going on by taking records. Now, obviously, people prefer to do controlled experiments unless it's too complicated or too expensive, as in the case of, well, telescope-related studies, for the most part, for what I hope are very obvious reasons. Now, the controlled experiment has the independent variable, or independent variables, because most experiments have many independent variables, although people do try to control all of them except for one at a time. So, the independent variable is controlled by the researcher, and they observe what happens to the dependent variable based on the independent variable. So in other words, starting conditions, result. Now the second type, the observational study, is, well, it's important that you do not affect this object. Even though we know that for any of us who like philosophy or who like scientific trivia, we know that according to the observational theory of, or whatever it's called, the Schrodinger's cat example is that if you cannot observe whether the cat is alive or dead, then at the same time it is both. This, of course, predicates on the idea that the universe itself doesn't bother observing the cat, which is a little bit off, given that, well, gravity has an infinite range, and any changes in the mass distribution of the cat, or the box, will eventually propagate through local space, at least. Now, so, the third type of inquiry is a correlational study. Technically, this overlaps with both of the previous types, because I have done many controlled experiments, and in basically all of them I have to calculate for correlation to a certain expectation. Now, the correlational study is a matter of a relationship between a pair of variables, and if there's a positive correlation, then both increase. Or if one increases and the other decreases, then it's a negative correlation. If there's no correlation, then they're just random relative to each other. The correlation may be strong, i.e., all the dots can be, say, on a single line, or it can be weak. Now, we do point out that the correlations do not necessarily have to be linear. They do not have to be a straight line. They can be a curve, or even a parabola. Admittedly, this is not the best parabola, but, well, you get the point, I hope. So, if the correlation is strong, that means, well, this factor strongly affects that factor. The independent variable strongly affects the dependent variable. Or it could be vice versa. Why? Well, let's think about this. Say, food intake for people who all have the same amount of physical activity each day, or roughly the same, versus, well, pounds overweight. Well, I mean, these are going to be relatively tightly correlated because they have similar amounts of exercise, all of these people. Whereas if you selected a group where they all have different amounts of exercise and you use food intake per day, food energy intake, versus pounds overweight, well, you're not going to find a very strong correlation because of the other factors involved. And then, of course, there is a matter of randomness in biology and in other sciences as well. So, scientific investigation has a particular procedure. First, you need to get started. I mean, initiation, and, well, you need to make a plan, even though we know that according to many great men, actual plans never survive contact with the enemy, in this case, the randomness inherent in science. So even though we know that actual plans don't survive, planning is important. And then what do? Well, you have to actually do the experiment, right? After you do the experiment, you take the information, you analyze it, and then you have to present it to somebody. You have to sell your information to people, so to speak, to earn yourself, well, acknowledgement, recognition, fame, fortune, whatever. But we do point out that contrary to stereotypes, scientists are for the most part not loners. Scientists do work in teams, despite the fact that to make less people interested in science, because they don't need so much competition, the common media stereotypes scientists as nerdy loners, often enough. This is very much not true. If they are loners, they are very much loners by choice. For example, well, Nobel was very, very picky in terms of contesting patents for dynamite. Of course, some people got around it by using a slightly different formula. You probably know Alfred Nobel in terms of Nobel Prize. Now, that's actually it for this section. So really, the only difference from grade 9 is pretty much the addition of correlational studies.